Okay, so the first practical is actually to repeat the classical experiments by Sergei Winogradsky and also by Martinus Beijerinck. Okay, so this is the first Winogradsky column is a bit messy. So, but y'all don't have the chance to do it, but I'll just show you how usually how the students do it. Okay, do is usually what we do is can you see what I'm right? You can see what I'm drawing.
Yes, I can.
Yep. Okay. So usually you have a tray here, then we'll put the mud, okay, in the plastic tray. We add the water from the tap, and mix it well to create a slurry. Okay, so this is an important step. A slurry means it's muddy, it's liquid-like. Because when we are doing it, we will remove the particles like the twigs, shells, and other larger objects. We do this because we need to create a homogeneous soil layer. Okay, we don't want opportunities for the bubble to be trapped, or for any bubbles to be trapped, or for the chemicals that we're going to add into it, for it to be not homogeneous. Because if there are twigs, if there are solids inside, some of the chemicals might be trapped uh, at several hotspots and not evenly distributed, so it won't be homogeneous. Okay, so we create this slurry to ensure that the distribution of the chemicals are homogeneous. Okay, only once we've done that, we will prepare the Winogradsky column. Okay, so usually what the students will do is, from the PET bottle that they have, okay, we will cut it. Okay, we will cut it, and then this top, we use it as a funnel. So we will invert the top and use it as a funnel as we pour the slurry into the bottle. Okay, we will pour it about this 2/3, and then we will top it up with water, tap water. So when you're pouring the slurry, it's important that you do not trap any bubbles, because if you remember from the theory, you're supposed to create a oxygen profile, a gradient, oxygen gradient, where it's anaerobic here, microaerophilic here, and aerobic here. So this is how you do it. Then you will observe the changes over time. Usually we will do a full 6 weeks. Okay, and we will see every week what happens to it. For the final bottle, we will also cover it up with a saran wrap and make holes inside. Make holes in saran wrap. We cover it to reduce the condensation rate, but we poke some holes there so that the surface layer remains aerobic. So if you find that the water level has decreased over time, you can top it up, make sure that it doesn't dry up. Okay, if it dries up, then your whole column will spoil. Okay, so this is how you should be doing your Winogradsky column. Any questions about this?
No, doctor.
Okay, so just look through your practical schedule, and there are questions there that you should try to attempt to answer. Okay. So the next one is the experiment by Martinus Beijerinck, the isolation of Azotobacter. Okay. So what you will be supplied with is actually a conical flask with the N3 broth. Okay, conical flask with N3 broth. Of course, you'll be provided with a spade, and then a container to sample soil. So when the students sample the soil, usually I will ask them to sample the surface, fertile surface soil, and preferably fertile. So usually it's the ones that are brownish to blackish. Okay, not we want it to be fertile because we wanted to make sure that there are at least some bacteria there that we can maybe get it to grow here. So if you notice, you are asked to place the soil here, just a little bit of soil. Okay, it's about what, 0.2 grams, is it? Or just a little bit? Yeah, 0.2 grams into 50 mils of solution here. Why can't we add more soil? Wouldn't that increase the chance of getting nitrogen fixing bacteria? Why do we add so little? Anyone?
If we add too much soil, there's probability of other organisms also in the soil.
I'm looking for another answer. But even if there are other organisms in the soil, the media is N3, so only the only the nitrogen fixing bacteria will grow. So why do we cap it at 0.2 grams? And I mean, why don't we add more? 2 grams, maybe 10 grams?
Because if the soil is too much, then there's probability of air bubble could be maybe inside.
Good try, but that's not the answer. Anyone else? Good try. It's always good to hear students trying. No harm. Okay? You all should learn to interact more. But yeah. Anyway. Anyone? Okay, this is an N3 broth. The problem is if you add too much soil, it won't be N3 anymore because the soil itself has nitrogen. Okay? So that's why we have to strike a balance between the inoculum size and also the to be wary that too much soil will contaminate will enrich your broth with nitrogen. Okay, so that's why we keep it we keep the inoculation of the soil to a very small amount. Okay. So when if you read the schedule, you will also know that you are asked not to initially you can stir, then after that you're supposed to mix it up but after that you are asked to uh, just gently handle it gently. Okay, even when you after 1 week you will be asked to use an inoculation loop and take from the surface layer and then plate onto do a dilution streaking onto a end-free broth. Okay, so why gently? Why do you think I state gently there?
Because
We don't.
Aerobic microbe. So it will, because it's aerobic, it will prefer the interface. Okay, so that's why it will grow and as a surface film on the top. So you do it gently so that you don't disturb it. Okay, so that you can increase the chance of isolating the azotobacter that you want. Okay. So once you have your culture, presumptive, something that grows on the end. So once you have it, uh, then you will do a gram stain. Okay. Um, yeah, you will do a gram stain, and then you can also see under UV. It will fluoresce. Okay, some of the species will fluoresce. Based on the table in your practical schedule, you can determine the species of the azotobacter. Okay. So yeah, so look at the questions carefully and then you can answer them. If I'm not mistaken, the questions are located at the back, no? Where are the questions? Yeah, the questions are here. Okay, these are so the questions. So look at the questions carefully and try to answer them. Okay. Um. Okay. Maybe we should attempt the question. So for the question for the Winogradsky column. Okay, number one, why do we ensure that no bubbles are trapped? So I've already told you the answer. Uh, we need to make sure that the no bubbles are trapped because for the Winogradsky column to work, there has to be a dissolved oxygen gradient from the surface to the bottom. So if there are bubbles trapped, then you will have you won't get nice layers. Okay, uh, nice layers of microorganisms. So next one, how do the physical chemical variables change throughout the column? This one you have to look. So we use soluble cellulose as a form of carbon, but we use cellulose so that the carbon release is slower. We don't use sugar. If we use sugar, then the carbon release will be too fast. Then the whole column will become anaerobic. Okay, that's why we use cellulose slowly. Then the only difference is the calcium carbonate, ammonium chloride, and the dipotassium hydrogen phosphate. These are similar between B and C. So basically, it's to supply uh, carbonates, the ammonium, and also the phosphorus for the microbes. But the only difference is calcium sulfate in B and ferrum sulfate in C. That's why theoretically in C you should see a blacker form of column because the ferrum will react to form ferrum sulfide. Okay, so that's black in color. Whereas the calcium sulfide will be, when it's reduced, will be whitish in color. So there should be a color difference as you incubate it longer. Okay. But you can probably get more details on the net about the changes that occurs throughout the column. Okay, so number 3 is also similar to the uh, answer that I gave earlier. So can you relate the changes in your Winogradsky column with the chemicals that you added? Okay, so we should see different color schemes here. Of course, greenish cyanobacteria will be at the top. Then you should see some uh, maybe uh, purplish color also, depending on the types of microbes that are in your Winogradsky column. Okay, so for Azotobacter isolation, the questions are: do you think all the microbes growing in your enrichment culture and fixes? Explain. So we hope that they are all nitrogen fixers, but as I mentioned, the soil itself contains some nutrients. That's why for this experiment, you are supposed to incubate long. About you do a inoculation dilution streaking after 1 week, but you continue doing dilution streaking um, in the second week. Okay, on the assumption that on the hope that the all the nutrients in the soil will be fully used up by the second week, and only the organisms that can fix nitrogen are growing in your broth. Okay, so why do we not shake the flask? Why do we not shake the flask? Okay, why do we not shake the flask or disturb the surface film? I've already explained this. Then number 3, everything is everywhere the environment selects. Okay, so this, I'm sure you will be able to explain this statement, because when we have a nitrogen-free broth, only nitrogen fixers can grow. Okay, any other question before we move on?
Doctor, I actually have a question about the Winogradsky
Yeah.
experiment.
Mhm.
Why do we just tap water instead of like water? Because tap water they contain chlorinated water, right? So wouldn't this be
uh,
the loss of bacteria?
Adi, I cannot hear you clearly. Can you type it out in the chat?
Okay.
Sorry, yeah, your voice was breaking up. It might be the reception here. So the next experiment we'll do while we wait for Ali to type is soil respiration. Is Ali typing? Okay. So okay, what is let me see. Why do we use tap water? Oh, okay. Okay. Why do we use tap water? Uh, uh, okay. Tap water contains chlorine, but when you're mixing with the soil, I don't think the chlorine will affect much of the work you're doing, because one thing, uh, chlorine in water will evaporate. And second thing is chlorine that is detrimental would be captured by the soil particles, and usually it won't affect so much. Okay, that's why we also use tap water because it's easier. Because no point, and also sometimes the tap water also contains certain elements and chemicals that will help the bacteria to grow better instead of distilled water. Okay, that's why we use tap water. Of course, if you want to use distilled water, I don't think that there will be any problem. Uh, yeah. In fact, if you're doing a in-depth study in nutrients element cycling, then it is advisable to use distilled water. So at least you know what are the chemicals in your column. Okay. 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 Let's move on. So the soil respiration that is in your practical schedule is a simpler type of analysis because we use chemical titration and reactions to determine how much respiration is being carried out. So respiration process is where the CO2 is released. Okay, CO2 is released, oxygen is consumed. That's respiration. That's essentially what respiration is. So uh, there are two ways you can measure respiration. One is by looking at dissolved oxygen decrease, and the other is looking at CO2 release. Okay, so for this experiment, we'll be looking at the amount of CO2 that's released. Okay. So if you look at the clip that I sent you, the short YouTube clip, it shows you how to prepare the experiment. So basically, here in the first bottle, we usually have 3 sets of each. This will be your control, and this will be your sample. So usually test, control and test. So first thing you have to add your NaOH. Then you have to prepare your soil. So what happens is we will use the gauze. Okay, gauze. Can you see gauze? Then we'll put the soil here. Okay. And then we'll tie it up. Okay. So for your control, this one doesn't have any soil inside. Whereas for your test, it will have soil. So when you place it in, make sure that it doesn't touch the reagent. Okay. Then you cap it up. Okay, so this one has soil and this one doesn't. Okay. So what happens next? We do 3 times triplicates because there's a lot of deviation, variability. So sometimes your standard deviation that you get from these results tend to be high. So if you don't do triplicates or even more than that, your results will be quite unreliable. Okay. So it's better to have triplicates for this experiment. Is my voice clear?
Yes, it's clear.
Okay, because yeah. Okay. So we incubate this overnight, about 24 hours, and then we will measure. Okay. So what do we do? Once you are done incubating, you will remove the pouch. And immediately add barium chloride. Okay, immediately add barium chloride. Swirl well for both, and then you titrate with HCl. Okay. So, do you know what reaction? What is the reaction? What kind of reaction is occurring? Okay. So let's just say, so when you do this titration, of course, when you do titration, your indicator here will be phenolphthalein. So if you still remember your high school chemistry, okay. So this is your control, blank, and this is a test. Which one will have higher titrant volume?
The test.
The test will have higher titrant volume.
No. The control. The
control
control.
will have higher titrant volume.
Yes.
Okay, why?
Because when we because in the soil there's some organisms, so when the respiration occur, so that the natrium hydroxide will react with carbon dioxide and forming sodium carbonate, right? So that the pH also will increase, so that the control tube will have higher pH level, so that it need more what we call we need more HCl to 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 change the phenolphthalein color.
Very good. Does everyone agree? So, so this test has soil. So there is CO2 released. This test, no soil. Okay. Of course, there is ambient CO2 here, but the ambient CO2 should be the same for both. So this one will be there will be more CO2. So the reaction, how is the reaction going to be like? Okay, so let's see whether we can balance the reaction up. So when you have CO2, you will react with your sodium hydroxide to give you sodium carbonate. So you have to have. Does it balance? No.
Na2CO3.
Na2CO3. Oh
Yeah.
yeah. So this one will be 2. Then you have a H2O here. Okay, correct. Now it's balanced. Okay. Balanced, right? Yeah, it's balanced. However, this reaction is not stable. This product is not stable. That's why when you're done with the experiment, you remove your your pouch. You have to fix it with barium chloride. So that will give you barium carbonate plus 2 correct? Balance?
Yes.
Yes?
Yeah.
So, what we are titrating is the leftover NaOH. That's why, so when we are titrating, oh, can't see. So we are titrating uh, uh, okay. So this is what we are titrating. What is happening when we do the titration? So we are titrating for the remaining sodium hydroxide. So uh, this will have less sodium hydroxide remaining. That's why your titrant volume will be less for this, and this will be higher. Okay, so do you understand now, everyone? So the reactions are all, the equations are all can be balanced out. Okay, so I think this is one of the uh, questions set for for this practical. Okay. So you also have a part where you are asked to take the soil, okay, put it in aluminum foil. Okay, you're supposed to measure it, weigh it, and then dry it. So when you dry, it can be air dry. Air dry essentially means just putting it in the lab for about 1 or 2 weeks and just letting it dry by itself. Or if you can't wait, you can dry it faster at around 60 degrees Celsius. Okay. Usually we do it 1 week because the next week is when we have the same practical session. So this is your wet weight. So this will be your dry weight. Okay. So the difference between these two will give you your water content. Okay? The difference between these two will give you your water content. Water content is quite important in soil science because water content relates to porosity. So the more porous the soil is, usually it can hold more water. So it will be more fertile also. So of course, next is you will do a furnace. Place it in a furnace where you ash it. So usually this is about 500 degrees Celsius for 3 hours, roughly. Okay. We say ashing it okay, because whatever remains are the ashes. So what happens here is all the organic matter will be uh, combusted into CO2. So there will be a weight loss here, and this is your ash. So the difference between your dry weight and your ash is your organic matter. Okay. Okay. Do you all understand this flow?
Yep.
Okay. So when we are doing the soil respiration experiments, the students will be asked to collect the soil sample, measure these basic parameters, and then carry out the respiration experiments, and then try to analyze to test the hypothesis. So what kind of hypothesis can we test? So basically we can test the following. Does the soil respiration relate to water content? Does the soil respiration which means does the soil respiration increase as organic matter in the soil increase? So these are the hypotheses that we can test from the results. So what I will be doing is I will give you some data, and then you all try to do a analysis on this. Okay. So what kind of a test, statistical test, do you think is possible for this? So you want to see, does the soil respiration increase when organic matter increase? So what kind of a test will you do?
BOT.
No, statistical testing.
Oh, sorry.
Anyone? Statistics. First year, biostat. Anyone? No?
Innova.
No
I'm-
one tells you whether your samples are different, but it doesn't tell you relationships. So for relationships, you do a correlation. Okay, so correlate your respiration to your organic matter. Okay. So if the organic matter increase, your respiration increase, then you'll see whether the statistics is your slope is significant. Okay, so correlation. So if I'm I think it's Pearson correlation. Pearson's correlation. Okay. Okay, any questions?
No.
Okay, so if there are no questions, I think we'll end it. We'll end the session. So I'll give you the data, the raw data. Can you all help analyze? Try it out. If you all do it, you can send to me. I can help you check. Or give you comments. Okay.
Submit it via Spectrum, right?
There are no marks for this, so but I think you should do it because it might be tested. You may be tested on it. So I think you should do it. Okay. You can submit it to me. No, this one you can just do it normally via Google Classroom. The one I think you're supposed to do a report for me, right? It's okay. We do one step at a time. So I'll give you the raw data. You spend some time doing the analysis. I'll give you a deadline. Then when you're done, you just send it to me for checking. Okay? Then we can go through it.